now let's have a look at Module 6. Finally, we are here. Yeah, Module 5 just usually takes a lot of time because it's so dense and there's so much to go through. But I'm hoping you guys feel refreshed with all that Module 5 review. Please ask if you have any questions. So let's begin by looking at Module 6. So we're looking at 3 specific dot points. We're looking at mutations, we're looking at biotech, and we're going to look at genetic technologies. So we are going to focus on these specific questions. We're going to look at how does mutation occurs. How does mutation occur? We are looking at what are the possible outcomes of a mutation. How can we actually manipulate the knowledge of genetic change for our advantage? We're then going to look at what is the deal with the field of biotech. What exactly is biotech? We're going to look at what are the cool genetic technologies that we have developed and their uses. And then in the last part of genetic technologies, we look at the social, economic, and environmental and cultural implications of genetic technologies. So let's get into it. Let me just grab a quick sip. So our inquiry question is, how does mutation introduce new alleles into the population? So Module 6 is a really nice segue from Module 5, where now when we look at mutations, firstly we're going back to our processes of replication, translation, transcription, and talking about mutations affecting those processes. Okay, here we are. So first question we have here is that we're going to look at is, how do mutagens operate, the different types of mutations. We're going to talk about somatic versus germline mutation, and then talk about the effects of mutations on coding versus non-coding DNA, and then look at causes of genetic variation, and finish off by talking about population genetics. So we're going to talk about mutations, gene flow, and genetic drift. So here we are. Our inquiry question is, how does mutation introduce new alleles into a population? Remember, a mutation, firstly, is a permanent alteration to the nucleotide sequence of an organism's genome. This is where we are permanently altering someone's, or rather, natural circumstances or their own system leads to the alteration of someone's own nucleotide sequences, which then changes the proteins that they are able to make. So we'll firstly begin by looking at the mutagens. What are the things that actually cause these mutations to occur? And we're going to talk about electromagnetic radiation first of all. Electromagnetic radiation, as the name suggests, refers to the waves that are emitted by electrically charged particles. They interact and ionize atoms that they encounter, and there is a danger, and the danger of radiation lies in its penetrative ability. So electromagnetic radiations can penetrate quite deeply, and they can travel into the nucleus, and they can adhere with DNA molecules, and they can lead to the creation of free radicals. So it's not very good at all. And how does it actually cause mutation? So once it actually penetrates in, there's a lot of energy, right? Electromagnetic radiation, there's a lot of energy input, which actually may alter the way that the DNA is bonded, and it can change the chemical composition of the DNA as well. It can also lead to the rearrangement of the structure. So here we are, there are examples. So for example, ultraviolet light is an example of electromagnetic radiation, and it is radiation from the sun which, if encountered in high doses, in daily high doses, can be harmful. However, we do encounter it daily, and that's why we know that there is huge stress on the importance of wearing sunscreen, being sun safe, wearing protective clothing, because we know that there is an increased risk of melanoma from it, which is skin cancer. So UV light is harmful in high doses, and what it does is that the incoming photons actually excite the adjacent pyrimidines. So these adjacent pyrimidines, which includes T and C, so thymine and cytosine, so it incites them, and it then causes them to actually turn around and bond to each other through covalent bonds. So instead of the hydrogen bonding here, that hydrogen bonding is broken, so the C and the T can bond to one another. And on an average summer's day in Sydney, a person will acquire around 20,000 pyrimidine dimers. So it does happen. It is a very, very common thing. But, and there are lots of mutations that occur, but the thing here is that we do have our own repair systems that do repair some of these changes. Well, we'll talk about what happens if those changes aren't repaired. But this is the first mutagen that we look at, electromagnetic radiation. Now let's have a look at chemicals. Chemicals are also mutagens. We have radioactive agents, so agents like uranium, and they can release radiation, including alpha and beta particles, and they are going to be high energy particles, and they are going to they can include alpha and beta particles, which are high energy, and they can also include gamma waves, which is actually a form of electromagnetic radiation. And both of them can then, or all three of them can then penetrate the cell and interact with the DNA bonds, again creating structural disruptions. In terms of metals, so the metals that are mutagens include arsenic, nickel, and cobalt, and so being exposed to them for a long amount of time, it can affect the integrity of the DNA repair system. So we have our own inherent repair system that essentially stops the proteins from that basically fixes any mutations, or tries to fix as many mutations as possible. But if that repair system is affected, which is the case with metals, that they can affect the DNA repair system, they can actually stop proteins from being able to recognize base pair mismatching. So it stops that recognition of base pair matching. So any incorrect base pair matching, it stops that from being detected. And so as a result, we have that's overlooked, and that mutation goes unnoticed, and it reduces the fidelity of the DNA replication, so more errors occur. And nickel especially inhibits the ability for histones to condense DNA. So histones are really important because that's where they're wrapped around DNA, and they essentially protect our DNA. And if they essentially and if nickel affects the ability for histones to condense that DNA to get that DNA to be protected, it can then lead to the chromosomes being in a bad formation, and that can of course affect the protection of the chromosomes. Last one, naturally occurring mutagens. So naturally occurring mutagens includes viruses. So these are things that are out there in nature that can lead to mutations. So viruses are an example of this, and what viruses do is that viruses replicate by inserting their own genetic material into the host cell. So they insert their own genetic material into the host cell, and they disrupt the normal cell function. And by doing that, that can lead to lasting changes to an organism's genome. So viruses don't have the machinery to actually make their own proteins. So every time a virus, so they need basically another body to survive. Like they need a human body to survive. So they get in there, they're going to get into our cells, gain entry, hijack our whole transcription translation machinery to then put in their own genetic material, so that that material can be produced, so that their proteins can be produced. And these are some examples of viruses. So HIV, of course, which then leads to AIDS, hepatitis B, Ebola virus is another one, adenovirus, influenza, and bacteriophage. And bacterial infections, which is bacteriophage right there, what they do is that they induce inflammation. So they cause inflammation, and that reduces the efficiency of our DNA repair systems. And it also then leads to the increased in mutations. And a good example of this is, for example, Helicobacter pylori, which is a bacteria, and it has actually been linked to the development of stomach cancer. Remember, cancer is that unhindered replication of the cells, right? And what happens is, if our DNA system is repair system is unable to repair that mutation, the cells keep replicating, and then that then leads to cancer. And in this case, there has been correlation between Helicobacter pylori and the development of stomach cancer. So now we know what these mutagens are. Let's talk about the effects of these mutagens. What exactly happens? So sorry, let me grab a quick sip. Alrighty. So here we are. We're going to now compare the causes, processes, and effects of different types of mutations. But we're first going to talk about point mutations. So point mutations, in terms of what causes them, firstly, they are caused by external mutagens, DNA replication errors, and DNA repair system malfunction. So point mutations are these mutations in the coding sequence. So they just affect, one or two nucleotides. It just affects the coding sequence here, and it can have these specific effects. It can lead to a silent mutation, which means we don't actually see it having an effect on the amino acid sequence that's produced. It can be missense, nonsense. It can lead to the creation of SNPs, which is a single nucleotide polymorphisms, which is the change in a single nucleotide. It can also lead to the creation of new alleles. So mutations are not always bad. They are important for variation, but they can be bad. They can be potentially beneficial, and they can be potentially negative. And just because a point mutation is small doesn't mean that its effects will be small. So cystic fibrosis is an example of that. So here we have point mutation, an example of what actually happens. This is the mechanism, and this is the effect right there. So essentially what happens is that in point mutations, this is a normal codon sequence, right? So we've got, let me use a highlighter. Okay, so we've got AGC, we've got AAG, and GCU. We've got three specific sequences right there, and it produces these proteins, right? So these specific amino acids, which will then join together, form polypeptide bonds, lead to a polypeptide change, form proteins. So there are two types of point mutations that can happen. There can be substitution, or there can be a frame shift mutation. In substitution mutations, what happens is, as you can tell by the name itself, one of the nucleotides substitutes the other. So instead of the G here, the guanine, there has been a substitution, and there's been a uracil that has been inserted. And this, remember, this is happening during translation. So because of that uracil instead of the transcription has occurred. Translation is happening now, but during transcription there's been that change of uracil. From guanine we've gone to uracil, and because of that the code now changes to the codon changes to the anticodon, the codon changes to the anticodon, my bad. The anticodon changes to AUC instead of AGC, and that means that our amino acid will also change right there. Frame shift mutation can either be insertion or deletion. So frame shift means that it just shifts one over. So it's causing a downstream shift, basically, in reading the sequences. So how does that happen? They can either be insertion or deletion. In terms of insertion, what happens is that we have the ACG. So here we have inserted a C just before the G there, and that has, instead of changing just one amino acid, it's actually changed the whole sequence here. Same thing happens if a deletion happens. So we've deleted the G here, and that has also changed the whole sequence there. And its effects can be the following. This can be the effects of it, right? It can either be missense, which means that in this case here, the mutation edits the mutation which edits codon sequence to incorporate different amino acids in the protein. So there is a different amino acid that has been added in, and that is so that's an example of missense. This is an example of missense, and this is an example of missense. So a different amino acid has been incorporated in. Nonsense mutations, like the nonsense effect, essentially is when a premature stop codon is introduced. So when you look at this, you will actually look at a table that tells you, there's a table that actually tells you what the combinations of these anticodons, what amino acids that they'll lead to. And if you want to check it out, you can just type up amino acid translation table or something. It should pop up, and you could see, for example, that the codon here, AUAG, that is a stop codon. That is telling us that, okay, the process stops. And what it's done is, as you can see, it has prematurely stopped the process. It has prematurely stopped it, and it has prematurely terminated the polypeptide synthesis. A silent mutation, on the other hand, is when, luckily, yes, there has been a change here, but if you look at the amino acid table, you'll see that there can be a variation of sequences, of anticodon sequences, that lead to the production of one amino acid. So AGU also leads to the amino acid SER, that I don't know the full form of SER, but it leads to the production of this specific amino acid, and so does AGC. So it's like so there are a couple of anticodon combinations that will lead to the same amino acid being formed, and so as a result, luckily, there is no effect on the amino acid sequence. Okay, now that was point mutation. That's when when the mutation affects the nucleotide sequences. Point mutations is the first one we looked at. The second thing we're going to look at is chromosomal mutations. This is where the chromosome, the entire chromosome, is affected. And there are essentially two main causes for it generally. It generally is a result of errors that occur in meiosis, in the process where the gametes are being formed. So essentially, firstly, there could be an issue with crossing over, and if crossing over occurs incorrectly, that means that chunks of DNA can be screwed up, like they can move places, and that can lead to structural mutations, these mutations right here. Secondly, there can also be issues with sister chromatids being incorrectly separated. So remember, when we've got the sister chromatids that line up near the mitotic spindle, that line up and the mitotic spindle is attached to them, they're separated. If there's an issue with separation, or they may not be separated, or they may just be separated for like one side, that can lead to an uneven number of chromosomes, and that leads to mutation in the number of chromosomes. The effects of this are usually severe, more severe than point mutation, because this is where the whole chromosome is affected, so it can affect a number of genes. So these are the structural mutations that can occur. So when the chromosomes go to cross over, for example, it could mean that there's a deletion of a chunk of the chromosome. Inversion, they can like invert over here, as you can see. Translocation, a part of the chromosome here can translocate to the other chromosome, and duplication, it can duplicate as well. So it leads to so that's structural mutations. The second one is mutation in chromosome numbers. So for that, there's either aneuploidy or polyploidy. So aneuploidy is when the overall chromosome number of the offspring is different to the parent organism. So in the case of Down syndrome, the individual has 47 chromosomes instead of 46, because they have 3 chromosomes instead of they have 3 copies instead of 2 for trisomy 21, which is chromosome 21. So they have 3 copies of it instead of 2. In terms of polyploidy, what happens is that when an organism contains more than 2 sets of homologous chromosomes, so instead of having 2 chromosomes, they actually could have, for example, 3 copies of every homologous chromosome instead of the 2. And an example of that is triploidy, the condition. An example of aneuploidy is Down syndrome. Okay, awesome. And now let us talk about somatic versus germline mutations. You kind of would already be familiar with this specific concept from module 5. So, germline mutations, germline, it's going to affect the germ cells, the gametes, right? So, for example, these are the parental gametes. They're going to fertilize. There's a condition, let's say here, that there's a trait specifically that's carried in the mother's in the chromosomes here, in the egg, and so when fertilization occurs, there's an embryo, the entire organism is going to carry that mutation. And because the entire organism carries that mutation, when that organism reproduces, half of their gametes will carry that mutation, which means that depending on whether it's dominant or a recessive allele, the offsprings may have that mutation because it's affecting the germ cells, it's affecting their sex cells. On the other hand, somatic mutations is a somatic mutation. Just has somatic mutation is a somatic mutation. No, somatic mutations will only affect body cells. So what that means is here if you have a mutation in some cells over here, it's only going to affect this area. It's not going to affect the offspring of the individual because the mutation is in their somatic cells, it's in their body cells. The mutation isn't in their germ cells, it's not in their gametes, so then it won't pass on to their offspring. So there we are. All I just said. So germline mutations affects the gametes, and so changes to the gametes leads to changes to all cells of the offspring, and the offspring will inherit mutated information, and it can be caused by both external and internal factors. An example of that is cystic fibrosis. Somatic cell mutations are when mutations affect your somatic cells, your already existing cells, your body cells, and so that means that it's not going to affect the future generation. So example of that is skin cancer, where for example, if an individual has skin cancer, it won't be passed on to their offspring, because it only affects that individual's somatic cells. But there could be like genetic factors and stuff that means that the individual may be more susceptible to it, but that's different. Like they wouldn't have it just because the parent had it. Whereas in germline mutation, the chances of the offspring having the condition are pretty high because depending on whether it's dominant or recessive, because it's passed on through the generation. Okay, so now we're going to look at, and here we go. Okay, now we're going to look at the significance of coding versus non-coding DNA sections of the DNA. So remember, not all of the DNA is coded for by proteins. Not all of the DNA codes for proteins, my bad. So what we have here is that coding DNA, in fact, is very small. Like it's a very small part of the overall DNA. Like a very small part of DNA codes for genes. Very, very small. That's our coding DNA. The rest of it, we have junk DNA, we have enhancers and silencers, we have promoter sequences. That name might stand out because remember we talked about how in DNA replication and in transcription and translation we have promoter sequences that actually tell us when to or where to actually start these processes. We have introns, terminator sequences, and non-coding RNA as well. So here is the purpose of each. So genes are, remember, these mutations that occur to our genes, to a coding region, it can have an effect on the protein that is produced, and it can have a serious effect on cell function. Mutations that occur to the introns, they may have effect on splicing of genes. Remember how when we talked about post-translational splicing, we talked about how we take out the introns so that we are left with the extrons, coding DNA, so it can have an effect on the splicing of the gene. Promoters, mutations to promoter region can actually inhibit the ability for a gene to be transcribed, because remember, those promoters are telling us where to actually start the process, where to actually start transcribing. So if there's an issue here, that will have an effect. Oops. Oh, my bad. Sorry. Let me just quickly. forward. Oh, that's fast. Perfect, and my highlights are still there. Awesome. Alrighty, even though they're terribly done, sorry about that, but they're still there. So, we talked about promoters. Now again, terminators, same thing as promoters. Mutation to a terminator sequence can result in incorrect or elongated mRNA sequences because it doesn't know where to stop, so it can be incorrect and elongated. Mutations to enhancers and silencers, so these are the regulator regions. They actually tell, like, they enhance certain genes and they silence certain genes when we don't need them. So if mutations occur in this region, it can actually lead to some genes being over or underexpressed, and that can lead to either too much protein being formed or too little protein being formed. Non-coding RNA is when mutations occur to non-coding RNA sequences, and that can result in alteration to important molecules. So non-coding RNA is important for those RNA molecules that we talked about in the processes of replication, transcription, and translations, like, you know, tRNA is an important one, right? So if there's a mutation to that, it can affect the function of like tRNA and mRNA. Finally, junk DNA. Mutations to junk DNA, as the name suggests, won't have a huge effect, and they probably won't matter. So we can see how mutations, for example, I'll change color here, mutations to obviously genes, to introns, to promoters, to enhancers and silencers, to terminators, and also to non-coding RNA, pretty much to most of them, and, you know, almost all of them, mutations will have an effect. And that tells us that it's not the best, right? Like, it's not ideal to have those mutations. However, we will talk about how actually mutations, at the end of the day, they lead to genetic variation. And this is one of the key concepts here, is that the aim is for genetic variation for the continuity of species in a way. Right, and I'm kind of linking this back to module 5 here, but that's the key there. So we want to now talk about how through these different processes genetic variation is achieved. So mutation leads to the creation of new alleles through point and chromosomal mutations. Even if those alleles may not be the most beneficial, it still leads to the production of those alleles. Meiosis is when those alleles are recombined and sorted in random independent ways. So remember, we have crossing over, we have random segregation. So the way in which those chromosomes are separated is random, and also the way in which they're sorted and separated is also random. So that's independent assortment and random segregation. Crossing over, really important. You know these three processes in detail. Like, I would add that to my notes. And finally, fertilization. So this is where these variable alleles from two different organisms are brought together, and they interact in new ways to lead to the creation of new alleles and new phenotypes. So there is an uncontrollable combination of a number of possible gametes, and the interaction of alleles to produce phenotype also leads to genetic variation. Awesome. Now let's have a look, and I know it's a long dot point, but I believe this is one of the last parts of this dot point. So let's have a look at the effects of mutations, gene flow, and genetic drift on the gene pool of populations. So here we are. So a mutation, remember, is a permanent alteration to the nucleotide sequence of an organism's genome. It leads to the creation of new alleles. It can be favorable, and it can also, it may be favorable and subsequently selected. Gene flow is the transfer of genetic variation from one population to another, leads to changes in the frequency of alleles, and it can affect the distribution of genetic diversity. And finally, genetic drift is when the frequency of alleles within a population changes because of either disappearance of particular genes, and that could be a result of natural selection over time. So, for example, traits can become more or less prominent, and it can also lead to evolution of species and creation of new species as well. So, when we talk about genetic drift, let's just talk about a couple of causes. So that and this is kind of like a bit of revision from year 11. So bottlenecking is a process in which there is a abrupt reduction in the number of individuals. It could be due to natural causes, a sudden severe selection pressure, and that leads to a loss of diversity. The founder effect, of course, is when a new population is established by a small number of individuals. So they separate from the main population, they establish a smaller population, and it leads to a loss of genetic variation within the new group, and it can also lead to new speciation events as well. Evolution, overall, remember, is the change in a species over many generations. It results, and as a result of natural selection of favorable characteristics. And remember why we link this back to evolution. It's because we're talking about mutations, how it leads to genetic diversity, and how it can then lead to these specific processes. Remember, variation occurs within a population due to natural selection. My bad, right there, natural selection. Environmental pressure is applied to population, and phenotypes best suited to the environment, to the changed environment, will survive, and surviving organisms will reproduce. So this is telling us that genetic variation is super important, and I'm trying to draw a straight line, but that's not working. So genetic variation, GV, really, really important. And I want you to remember this because we're going to come back and link this back to biotechnologies and genetic technologies when we discuss that. Here is a practice question from the NESA's sample paper. We won't go through it because we're a bit tight on time, but what I will do is I'll leave that there, and of course remember you have access to the slides, and yeah, you have access to the slides to have a go at it. And this is for, like I said, this is from the sample NESA paper, so I think you'd also find a little explanation for each of these points as well in that sample paper. So if you just type up biology additional questions, sorry, yeah, not sample paper, my mistake, NESA additional examination questions, biology HSC, or something thereabouts, you'll get access to this. It's like an 80-page long document, and there are a range of questions on there. I might put it in the chat when we get up to this point in the recording, so feel free to remind me if I haven't already. So yeah, I'll put it in the chat, but you can access that, and it's got a little explanation as well. What I will do though is I'm gonna move on. Actually, let's do this part together. So here we have a practice question. So compare the processes and effects of point mutations and chromosomal mutations. Include examples in your answer. So sample answer, we can talk about essentially begin by talking about the fact, like, what mutations are, right? Define what mutations are and how they occur. So here we go. All mutations are changes to DNA. They occur during DNA replication, during mitosis, and during meiosis. So we actually identify what these processes are. In biology, always identify and give definitions for the terms in your response before you go ahead. Definitions are really important. We then have our first couple of paragraphs on point mutations. So as you can see here, it's not just one big chunk of paragraph, and that's because usually when you write a big paragraph, in cases like this for like an 8 marker, it can start to lose meaning, it can start to lose structure. So always recommend doing like smaller paragraphs. So here we are. Point mutations are changes that occur in a single nucleotide. These changes can be substitution with the wrong nucleotide, and extra nucleotide being added, so that's addition, or insertion, or a nucleotide not being included, which is deletion. So we've talked about 3 types here. So we've talked about point mutations, and how that's either substitution or frame shift, which includes insertion and deletion. It is possible to have multiple point mutations along a chromosome. The order of nucleotide bases determines the protein that is produced by the cell. So we actually link this back to the effect. We have to actually talk about what is actually happening here. The point mutation may have no effect on the protein being produced, as the change may still enable a triplet code for the same amino acid, or change of one amino acid might not have a significant effect on the resulting protein. So here we are. That is, can you identify which type of effect they're talking about here? It may have no effect. And so they're talking about the silent effect here. So that's silent S. The point mutation may mean that the triplet code initiates a stop sequence, which in which case the protein will not be produced. It may mean that a range of proteins are not produced at all, or the greater quantities of proteins are produced, and that will be nonsense mutation. And finally, frameshift point mutation is caused by an addition of deletion, is caused by an addition or deletion. Every triplet on the DNA after the point mutation is affected. This can radically change the protein product of the cell. And this is just a quick little summary on point mutations. And then we talk about chromosomal mutations. And remember, it's asking us to compare, right? So we actually have to link it back to the process and the effects. So here we are. Chromosomal mutations may involve large sections of the chromosome breaking off completely, or breaking off and reassembling in reverse order. So here we go. We are identifying those terms. Completely is deletion, inversion, breaking off, and adhering to another is translocation. These breakups of chromosomes may move genes to a new loci, and that's just moving them to a new location, and can break up genes by splitting the chromosome in the middle of the gene sequence. These types of chromosomes changes can radically affect cell activity. Chromosomal mutations also include non-disjunction of homologous chromosomes and anaphase. So we've talked about chromosomal mutations, and we've now talked about the mutations in the number of chromosomes as well. At anaphase, so here we are. There we go. So number of anaphase resulting the cell in many or too few. These can have radical effects on cell activity and the organism. And then we compare both point mutation and chromosomal mutation can cause disease. For example, point mutations can cause cystic fibrosis, sickle cell anemia, and chromosomal mutation can lead to Down syndrome and Turner's syndrome as well. Both kinds of mutations have also generated new alleles, which in some cases has been adaptive and contributed to evolution. So we kind of also want to talk about the fact that it's not always all negative. Make sure you know examples of each of point mutations and chromosomal mutations. Really, really important.